Hey, good morning. I'm Dr. Joshua Granier. And today, what I want to talk about is a piece of concrete evidence that connects a specific bacteria in the mouth with cancer. And this is based on 2024 Nature study. And I'm going to go into the foundational understanding of what's going on in that study. And then we're going to dive a little deeper, hopefully expand our understanding of how the mouth actually works. So, first of all, we're going to take a journey here and look at how complicated the oral biome actually is and understand how many microorganisms play a role and how they can get out of balance and various ways to start to try to understand what's going on. There really is an ecosystem, over 700 species of viruses, bacteria, fungi, protozoa, which are like large cell organisms with specific membrane-bound organelles, and there's billions. So it's incredibly complicated. Perfect condition for these types of living things to actually grow in your mouth based on temperature, moisture. And the amount of bacteria, the amount of life is only second to the gut in concentration. So remember, it's not a solitary thing. It's not one living thing. We really do team with life, microbial life. It's astronomically complicated. It's very important to try to abstract what's going on in ways that we can start to wrap our heads around. And one way is to look at the benefits of these different types of living things, whether they're symbiotic, whether they help us. And there's some mention that some microbes in our mouth are actually able to help process nitrogen, which can benefit us. Many simply live in our mouth, and there's actually certain species that will compete with others that are maybe more pathogenic. So there's a competition, and pathogenic, which are the ones that hurt us, the ones that cause periodontal disease, and also the one that we're going to be talking about very soon. So another way to try to understand it is there's a core amount of microbiome that everybody seems to have that are very predictable from one person to another, and they're presumed to be found in healthy individuals and unhealthy individuals. Now, the mouth is a gateway to the body. So anything that goes in your mouth is going to change or affect the microbiome. So if you smoke, you have too much sugar, too much acid, alcohol, dry mouth, anything changes your saliva. If you have diabetes, you have more sugar in your saliva, low calcium in your saliva, all these things affect the type of microbiome that you're going to have in your mouth. And very often it's going to make it dysbiotic, where we're going to start to see instances of oral disease and perhaps disease elsewhere. And presumably, and these are all broad generalizations, just ways to try to understand what's going on. They say balance is key, where if you have a good thing, it's symbiotic, you don't have an overgrowth of certain different types. Whereas once that starts to get disrupted, polluted, then you're going to see dysbiosis. And again, huge, just an attempt to generalize. Remember, there's many microbial niches in the mouth, and certain types of these organisms are designed to function in different areas. So the teeth, the hard surfaces of the mouth, can harbor calculus. It sticks to them. And plaque is driven by the structure of plaque is actually driven by the bacteria itself once it gets large enough, which then drives calculus. So these things can grow in ways that benefit calculus, taking on certain types of structures, fractal-like structures, broccoli-like structures, in other words. And then you also see those same types of things in strep mutanes when you have cavities. So not only can it grow out, but it can actually grow into the tooth and create an environment within the tooth where bacteria can start to thrive. And those also have a certain type of continually advancing structure. So it's very interesting stuff, but it doesn't stay there. It can actually invade the soft tissue and start to fight our immune system and cleave cells within that soft tissue. Particularly, we're talking about in periodontal disease or necrotizing type periodontal diseases, especially. There's many examples. This allows a high degree of specialization where different types of bacteria, there's different types of food sources, there's different types of interactions, and this community is able to work together. They're not there by chance. Now, there's a specific architecture where it is based on dental plaque, which is not just food, but it is a highly sophisticated living structure with a biofilm. It really is a city built by bacteria. And we're going to get into how they do that to a degree. How can it go from a clean tooth to this multi-species community? We're going to get into that. So first of all, everyone knows there's an acquired pellicle. You brush your teeth, immediately various proteins from the saliva are going to start to stick to that tooth, forming something called an acquired pellicle. And this may have some protective instances, but first of all, what you're immediately going to have is pioneering species, strep mutans, these gram-positive bacteria are going to start to stick there. And that's just the first stage, these sticky bacteria. Because after that, other colonizers like Fusobacterium, Nucleatum that are very sticky are going to then stick to that and start to grow it out even more. And they start to secrete a glue-like extracellular matrix, which is made of polysaccharides, presumably from sugars and foods. And it also helps form that matrix. Now, if you let it get big enough, and this has been shown, it will start to communicate Corphoronus gingivalis with something called corium sensing, which is an incredibly sophisticated signaling molecule, not just within one type of bacteria. Many different bacteria can communicate, and it will create a concentration gradient. So they'll know the relative concentrations of different types of bacteria. They can even make certain bacteria commit cell death and help grow that complex even bigger. And they can respond to acid attacks. They can respond to a variety of things, and they can grow certain broccoli-like structures that are very good for absorbing nutrients. So they start to almost come alive and become very sophisticated. And many people don't go to the dentist and you continue to do whatever's feeding. It will continue to grow essentially indefinitely. And also there's a microbial succession where as things start to develop, you have more bacteria. You will see more instances of periodontal. And there's certain trends that we see. We see certain things start to drop off. The overall amount of bacteria starts to drop. Aerobic bacteria, especially periodontal disease. In caries, you start to see acid starts to drop, the pH starts to drop. And overall, you get a more specialized type. You get certain types of bacteria that come in late, that have more of a cleanup duty. And one such thing that you'll see in more advanced periodontal disease is something called Fusobacterium nucleatum. And that is a bridging species, very sticky. So you see it all the time. Remember, they've been evolving. There's been periodontal disease since creatures have been eating food and using mouths. There have been bacteria in those mouths. They're incredibly specialized at doing these types of things. And they don't just float around. Once they're shielded by a matrix or even a calcified matrix, as in calculus, once they find specific areas, or even your gum tissue or your furcations can help shield them, they become incredibly resistant to antibiotics. Stuff like mouthwash just doesn't really work anymore. So they have a protected environment, nutrient channels, so they're able to feed better. They're able to capture more nutrients from your own saliva as you eat. And this is a continued and vicious cycle. The Streptococcus mutans is the primary driver of dental caries. And what that's able to do is survive and encourage lower pHs. So it's able to survive at a fairly low pH. And you will see biofilms that are driven by this, and there's many others, you'll start to see cavities. It will start to dissolve the tooth. And environments that are already acid heavy, if you drink soda, if you drink even non-sugary drinks, are especially able to cause an overgrowth of this. You'll start creating a vicious cycle, and then it will start creating a home for itself within a tooth, and eventually within the nerve of the tooth, if it's allowed to get deep enough. Cavities. Fusobacterium gingivalis is the key microbe in periodontitis, gum disease. Now, it is the key because it drives corium sensing. And there's other bacteria that may do the dirty work of actually causing inflammation and whatnot, the red complex, but this is often seen, and there's many other key bacteria that are seen in certain instances. And just to say, it's actually a lot more complicated than I'm even making it seem, because those 700 different species have many subspecies, and they're able to swap genetic information. Corium sensing helps with that. So again, you may have many different subspecies of the same type of bacteria that are wildly different and maybe evolved to work in just you. Assuming you're a smoker, you may have certain types of subspecies and bacteria that function very well in that specific environment. One of the reasons is there's really no way to separate the two spheres, because people that don't take care of their mouth probably don't take care of their body. If you don't think that brushing and flossing and eating well and avoiding soda and not smoking worth it, chances are you're going to have a pretty yucky mouth, and you're probably not going to be that healthy too. It's very difficult. Whereas if you take care of your mouth, you'll probably also have quite good health as well. So it's impossible to separate the two. And I think mostly it flows back the other way, where healthy people don't tend to have dental problems. But there's becoming more and more evidence. This 2024 study in Nature is going to show that there are instances where things that affect the mouth can actually affect the body in specific ways. And this oral systemic connection is real, but it is difficult to pin down. So the pathways, everyone knows, people talk about leaky gums. We see it all the time, where gum tissue on people with periodontal disease or calculus bleeds instant. It is essentially ulcerated. So that means bacteria will continually end up in the bloodstream. And I've seen bacteremia even in healthy folks, and it causes severe heart issues. And it was traced back to a oral bacteria, even an ordinary one. And there's a wide amount of science where they're seeing connections between rheumatoid arthritis. They're seeing gingipan, specific proteins found in periodontal disease, found in Alzheimer's. So the story is starting to evolve. And especially when you look at severe necrotizing types of periodontal disease, you can literally see a band of irritated tissue, presumably because there's bacteria living around it and affecting it and causing a severe amount of inflammation. So direct entry is a real thing. And you even see, surprisingly, even after you brush your teeth, you will see an increase of bacteria. And obviously this cause is inflammatory mediators. You can track these, but there's probably more to the story, where if you're seeing inflammation, you're probably seeing an actual concrete effect of that bacteria within the body. It's probably living somewhere or multiplying or doing something at another part of the body. And then the inflammation is secondary. But it's difficult to pin down by looking down a microscope what it's actually doing. It goes both ways, where people that have cardiovascular disease perhaps are doing things to their body, that they're not eating as well and exercising as well, that affect both systems, both their mouth and their body. But it also may flow back the other way, where you're seeing tartar of the heart. You can actually see certain things that maybe cause tartar in the mouth. Maybe you're doing a similar thing in blood vessels, and they can easily get there. And you start to see connections. Bladder stones, there's bacteria found in those that may be somewhat related or a distant cousin of the same things that causes calculus in the mouth. The process is there. And obviously these associations, stroke, diabetes is huge, even adverse pregnancy outcomes. Again, two-way street, hard to discover the truth, but it may start to flow back the other way. Interestingly, there is an association between poor oral health, poor oral hygiene even, and increased all-cause mortality, which is incredibly strong. It's very bad for you, where having a very unhealthy mouth is shown to be worse for your oral cause mortality than even smoking. So again, multifactorial. So the question is, if bacteria can travel to maybe influence the heart, is it plausible that they could maybe travel to the colon and develop cancer? And that is what we're going to talk about today. So let's go into part 2. Fusobacterium nucleatum. I've talked about this bacteria before, and it's really essential to the development of periodontal disease. It's highly adapted. And what is, it's very long, it's very thin, and it's very sticky. It is the master connector. It really does. I mean, it stands apart. And it's been often seen in oral disease, but now we're starting to see it in colorectal cancer, a completely unrelated area, different people, different scientists, but they're still seeing it. Pops up again. And what it is a gram nectarum bacterium, a broad shape, very thin, and it is anaerobic. It can't survive in oxygen, which means you're not going to see it on your tooth structure. And unless you have periodontal disease, you start to lose bone, and you have this packet that is essentially full of food debris and water. There's really no air that can get in there. And Fusobacterium nucleatum is able to survive there. So periodontal disease, you see it all the time and is related. The more peri you have, chances are the more Fusobacterium nucleatum you have. And remember, these structures it doesn't just burrow deep, but also if you have calculus, it starts to grow out. And then the deeper part of your calculus is also anaerobic. So it can actually survive in exposed areas of your tooth structure. if you don't go to the dentist and get it cleaned up. Now, it sticks to all types of oral bacteria. So your initial colonizers, your strep mutans, start to link it together, and then your late stage colonized Porphyromonas gingivalis can then start to bind to it. And it provides a lot of structure to your biofilm. It really drives it. It's a stable hub. Interestingly, oral bacteria, once it really starts cooking, can start to swap genetic material, which is why there are so many different subspecies that may have different types of qualities to them. And some of these things may be extremely pathogenic. It creates its own environment where it can thrive. So the analogy, it's a city. It's almost like a single organism that will start to thrive and start to structure itself in a way that will thrive in what is most likely a dysbiotic environment in your mouth. And they're starting to see this. Hey, why is this found in colorectal cancer? And it's a common resident. Almost everyone has it, but very rarely does it travel to the lower gastrointestinal tract. If you test healthy people, you hardly ever see it in their stool, but you see it in the mouth all the time. Interestingly, it is very much enriched in the tumor itself when compared to adjacent samples. So there's more than just an association here. There's suspicion that there could be a causation here. The higher the load of nucleatum, or FNA, as we'll start calling it, the more aggressive the disease, the more likely it is to recur, the more likely it is to metastasize, and ultimately your survival is going to be worse. So you're consistently seeing this association. It's bad on every single level when it comes to colorectal cancer. So this central question, is this a correlation or a causation? One idea is it an opportunistic thing? It just hitches a ride, the tumor's already there, and it just somehow ends up there. It's not really causing the trouble. Or is it the driver, where it is actively growing, just like it does in the mouth? When you think about how it grows in the mouth, where it's not the only type of bacteria, but it is a key driver. Its stickiness allows a lot of other, even more pathologic bacteria to do its thing. Maybe it's working more like that, where you're having a very pathogenic biofilm, obviously pathogenic biofilms in the mouth. Maybe there's one in the colon, and it's a key piece. It's been very hard to study this type of thing. So let's set the stage a little bit. How do you dive deeper? Where is it a bystander? Is it a key player? How do you start to tease out what's actually going on here? So first of all, this study by Nature, 2024, Zepeda Rivera, really looked at the genome of Fusobacterium nucleatum and start to tease out what's maybe going on. Tell more of the story. And what they found is, again, many different subspecies, but there were two specific clades, C1 and C2, that started to pop up over and over again. And what they found is, by looking deep, that there's a diversion. It's not just Fusobacterium nucleatum, but there are specific different families. You can look under the microscope and see a difference in these two different strains. So found that there were 55 tumor strains, 80 oral strains. And this is fairly typical. If you sample almost anyone's mouth, you'll see 80 different oral strains. It's really that numerous. They did sequences and looked at the genome, and what they discovered is that this subspecies belongs to a specific, when you look at tumors, they're highly concentrated around a specific subspecies. It's not just Fusobacterium nucleatum altogether. It's a very specific one. And it's very clearly bifurcated into two distinct groups. And they named them clade 1 and clade 2. And they were able to see very similar, nearly 91, 93% identical, very similar. And they were able to line up, track where the mutations actually happened. And what you start to see is, again, everything's found in the oral cavity. It's equal concentrations. There's no real preference of one over the other. But when you looked in tumors, C1, C2, specifically FNA, Fusobacterium, C2 clade, takes off. It is found in incredible statistics. This is an incredibly low p-value. And its close sibling, C1, doesn't do that. And associations are wonderful. P-values are wonderful, but they don't tell the whole story. So, of course, the Nature study, they dived a lot deeper. So what is so special? Why is it that C2 does so well in the colon? Why do you always find it? And they looked a little bit deeper. What is the culprit here? So when they identified it, they looked at his genome and they looked at certain tools. They don't understand everything in the genome, but they're able to say, hey, this gene codes for this type of protein, gives it this specific ability. And what they found is there's certain abilities that C2 have that logically, if you were making a bacteria, you would give it to allow it to travel to the gut and wreak havoc. So it has an expanded genetic arsenal. So much larger accessory genomes. These are the types of genes that it's able to take on. Remember, corium sensing plays a role. These genes, these bacteria are able to take on abilities from their neighbors. And that is really what we're seeing here. So if it wants to go from the mouth to the colon, it has to be able to survive the stomach. And they looked at that. It's got a GDAR system, GDAR, and C2 has it. Very special. Glutamate dependent acid resistance found in C2, not found in C1 or other types. Now, they looked at these and they simulated pH, and no surprise, C1 was killed very quickly by stomach acid. C2 survived it for 60 minutes, which is plenty long enough to survive the journey to the stomach. So it's an acid-proof vest. And that's just not it. It doesn't always cause cancer. And it's not only found in people that have cancer bacteria in the mouth. So you can find Porphyromonas gingivalis in the gut sometimes. And it will happen logically. Say you eat something and you get sick and everything just passes right through. Your stomach acid is very low. It doesn't kill for whatever reason. And a variety of reasons, you may end up having bacteria go from your mouth to your stomach. Where it would normally get killed, maybe you would just have a lot of bacteria in your mouth and you have digestive issues and it passes to your stomach. So it will spike up in the stool. It's not like it's low. It will low and then it might be really high and then eventually things will take care of it. Sometimes it can linger and cause tumors, but it's not the only time high levels. Once in the gut, and getting back to this, it's got to eat. So C2 has a genome containing a couple of operons, which are like clusters of genes coming together, switches where, hey, we can turn this on and it will metabolize a couple of things, a thalamine and 1,2-propanediol, which are foods found in the gut, but they're only found in the gut. And C1, of course, doesn't have these. And there's really no reason for it to have them if it's specialized for the mouth. So this is a bacteria that's specialized for the gut, but it could have passed to the gut and then taken on various types of qualities, genomes that were found from bacteria there. Because bacteria are able to swap information like antibiotic resistance. So it may have passed through the mouth on a pathologic individual, maybe a sick individual, and then it was able to take on some of the specialty functions from the gut. And it has FAP2 adhesion. And what they're finding is it's very sticky and it has increased copies of genes that make it sticky, increased copies of genes that make it stick to other microbes, fusulin. So it's got also the ability to damage host tissue, degrade immune molecules. Very interesting. So it doesn't just survive, but it has virulence factors, the ability to really wreak havoc and multiply. So oral cavity bacteria comes to play. It's there. It's highly adapted. And it does the same in the gut. So you can look at it. It's able to colonize more surfaces. It's able to move through mucus. It's able to evade and combat host cells. It is a strain that is specifically evolved to do and survive the gut. The weight of evidence, obviously, it's already very strong. They found the association. They've told the story. They've got the rationale. And again, it's a nature patient. They didn't even stop there. They looked at what's going on with CR patients. They paired the samples. Obviously, they know that total F nucleatum levels are higher in tumors. It's driven by FNAC2, and we all know why. And there's really no association with C1. So it is the tumor of interest. It's the prime suspect. Now, if you look at the tools, obviously on healthy controls, you're going to see a lot more of it. You see it in healthy controls sometimes, presumably because it passes from the mouth to the stool continuously, to the gut continuously. And when you compare them, obviously, most Fusobacterium stays in the oral cavity. It does not. It's not its niche. It's not associated with tumors. It doesn't have acid receptors. Doesn't metabolize stuff in the gut. Doesn't have these virulence factors. The overall shape is different, and it doesn't invade colon cancer cells. Finally, the last thing we're going to look into is part 4, going from association to causation. We're going to look at proof. Does C2 actually cause it? Can we cause cancer by giving something C2? And you actually can. So they did it in vitro, and they mixed it with a line of colon cells, and they said the C2 was able to thrive in cancer cells much better than C1 does. When you start looking at bacteria that thrive, they start, especially when they start to really take off and start to divide very quickly, they start to adopt these extremely fast-growing broccoli-like structures, fractal-like structures, which is just a way for cells to aggressively compete for nutrients and also allow nutrients to flow through them. It's an incredibly aggressive growth pattern. Our cells don't grow like that, but cancer cells do. So if you're an immune system, you're trying to combat a bacterial biofilm, you're probably going to have to start dividing yourself in an incredibly aggressive way. And that could be one of the reasons why you start seeing cancer cells to this high degree. So there is a functional difference in vitro between the two. But they ran an experiment, this poor little mouse here. They found some mice that are incredibly likely to get adenomas, intestinal polyps, which are the precursors to malignant tumors. And they obviously had a control group, a C1 group, and a C2 group. They compared them all together. They went head to head, and they wanted to not just compare no bacteria to bacteria, but really pit pathogenic suspects against its closest non-pathogenic relatives. So, and what they found is that, of course, the control wasn't non-zero because this type of mouse is predisposed to adenomas. So they saw some adenomas in this group, but it was much, much different. The group that was given C2 regularly, orally, by the way, had a vastly increased amount of adenomas within its gut. Very interesting. So that suggests that was the limiting factor. It comes down to C2. So how does it do it? It's creating an environment for itself. It's not trying to cause cancer, but it's causing an environment that leads to it. Manipulates the surrounding metabolites and terraforms the gut environment. And indirectly, that makes it much more hospitable to cancer, which are essentially fast-growing cells that belong to us, typically, and have lost their ability to stop dividing. And then they start to metastasize, meaning little bits of it can break off, and they will just continue to divide elsewhere in the body. And it is causing them to do this, our own immune cells to start doing this, or surrounding cells to do this. And perhaps it's by causing disruptive stress, oxidative stress, which is mutating these surrounding cells and causing them to grow uncontrollably, causing free radicals. What is it doing to these cells that stop them from working correctly and being identified? So obviously, you're going to see increased pro-inflammation. Our immune system does not like this stuff. It identifies as something bad. Our body does not like this. And you're going to see certain types of things that are known to promote cancer cells. So certain types of precursors on a low level, and very hard to predict how larger systems are going to behave based on metabolites, but you get indications that something is going wrong when C2 is around. And it starts to paint the picture that it is more than active. It doesn't just arrive there and live and indirectly pass through. It is living there, and it is fighting to stay there. And it's creating a hostile environment. And to me, it seems unlikely that it's doing it on its own. If you look in the mouth, the most virulent ones are ones that are able to communicate and work together. So I expect FNAC2 is not working alone. There's probably several others that are found with it, and it's just a piece of a larger puzzle. And again, it's an active subject. So what do we do about this in the future? So how do we fundamentally reframe the connections between the oral cavity and the gut? What profound implications for colorectal cancer prevention does this have? And maybe not just colorectal cancer prevention, maybe other types of cancer prevention, maybe even heart disease. So we're starting to understand there's a mouth-gut axis. And I think saying mouth even gives the mouth too much credit. It's really an understanding of the incredibly complex bacteria and systems that are easily observed in the mouth are probably those disease processes and are fairly well understood in the mouth are probably happening other places that are maybe not so easy to see. Oral bacteria enters the circulation through bleeding gums is likely true, but the fact that it can also pass to the body through the gut is very interesting as well. If you take a step back, you look and you see the C2, obviously it's found, it has mutated some, it's created a very unique ability to divide. So obviously, as an organism, it's very successful. It's evolved a certain mutation, and that mutation must be giving it an advantage somehow. And what is the advantage to surviving in the gut? How does it then spread to other people? Presumably, if it's found in your stool, it's probably getting spread via an oral fecal route. And that's a very common route of transmissions for lots and lots of foodborne illnesses and stuff like that. So is it possible that it's being spread? People not washing their hands, people with, if it happens to multiply in the gut, is it then getting spread back to somebody else? And taking a step back, the basic picture, C2 lives in the oral microbiome. And this is hard to show definitively, but it does live in the oral microbiome continuously. It's continually ingested, and can survive the journey. And it can then colonize the gut, and it continues to colonize the gut. And unfortunately, it's virulence, its extreme growth, its pathogenic potential allows it to cause cancer, and it seems to cause cancer. So the mouth may be a reservoir for this gut pathobiont, and C2 is the quintessential example. And with 700 species and over 700 many subspecies of those, it becomes very difficult. We don't even understand the full genome of C2, of what else it does. We don't understand a small piece of it. So this moves to oncology, maybe other diseases where do we test stool? Do we start looking for biofilms? Do we start looking for fractal structures elsewhere in other types of diseases? AI, and everyone talks about it, but when you look at how successful it was in protein sequencing, in determining protein structures, what it was able to do is take various, somewhat disparate pieces of data and not just optimize one, but optimize different types of systems all at the same time to create a full story of what's actually going on, or sufficiently a full story, which is something that a human can't necessarily do. We can maybe look at one thing. And this could be an option because we're looking at a sheer number of bacteria that's astronomically high, and not just bacteria, but viruses, fungi. We're looking at aquarium sensing, algorithm, a structure, and perhaps it may be necessary to take different types of data all together and see if we can better understand other types of bacteria that are probably doing this. Because if there's one or two or three, there's probably many more. And perhaps we can specifically target different types of antibiotics. Maybe not just target FNA, but target its ability to stick to other cells, target its ability to talk to other cells, or other cells to talk to it. Can you make a virus that specifically makes the environment more hostile to C2? Do we screen for it? And obviously, just by carpet bobbing, it's not going to quite work. It's already in an incredibly structured biofilm already by the time you have Fusobacterium nucleatum. So simply taking oral biotics probably not going to help. So how do we actually look into it? How do we target it? How do we model its travel? And that is very exciting and hopefully something that we can start moving towards. But first, we need to start looking for these types of oral bacteria elsewhere. So what does this mean for you? For science, I think it's very exciting. But for you, I think you just need to wash your hands, go to the dentist, take care of your oral hygiene, believe that you could make a difference in your own oral hygiene, and not just your oral hygiene, but make a difference in your health. Brush your teeth, eat well, quit smoking, lower that bacterial threshold in your mouth, floss, water pick, use a tongue scraper. It all helps. And exercise, eating, and that's going to lower your risk for not just colorectal cancer, but everything all together. It's, in many instances, cancer is not a random pattern. It's specific to a chain of events that almost seems random, but really is not. Yeah. Wash your hands, basic stuff, and understand that yeah, there is a transfer. Your body is a single system. Wash your hands. And remember, types of bacteria don't just travel for you. They also travel from you to your whole family. Like kissing leaves your mouth, travels very quickly to other people. And if you smoke, have periodontal disease, there's a good chance, and your kid smokes those periodontal disease, there's a good chance they're going to have a similar microbiome. So our mouths are complicated. They are an ecosystem, a living species that is doing what it can to survive. Some of the things it does make logical sense if you look at it from that point of view. Anyway, thanks for sharing. I'm going to be releasing the transcript, and please share. Thank you very much.